How many people do we have? All right. Do we want to give a few minutes for anyone else to arrive, or should I get going right away? All right, let me get my screen shared here.
Hello.
Hello.
I just wanted to come to thank you for joining.
Thanks
Give you
for
a
having me.
yeah. So yeah, I wanted to give a big thank you to Jacob Matthews for joining us all the way from virtually from another part of Toronto, Rotman Research Institute at Baycrest, where he's an MRI specialist. And I was so happy that he was willing to actually give us an hour introduction to MRI. So actually, with that, I will let you take off because I think you have a plan.
I do have a plan. Yeah.
Awesome.
All right. Thank you so much.
Cool.
So my talk today is going to be quite broad. I'm going to try to give you just kind of a big exposure to a lot of the different things that you may encounter in MRI and MRI analysis. I'm going to start with an intro to MRI, just kind of where our signal comes from, go through a bunch of MRI sequences, no sequence diagrams. I'm just going to show you some example images and talk a little bit about what kind of contrast they're using and what that can tell us. Because functional MRI is such an analysis heavy and critical part of neuroimaging these days, I'm going to do a whole little intro to functional MRI, including a little bit of analysis, not using any specific tools, but just giving you kind of an overview of what's typically involved, along with a few kind of more modern sequence options and analysis approaches that you might take. And then also, if I have time, give you a few ideas of some other data types you might encounter while analyzing MRI data, just some things that are possible to use simultaneously in the MRI. So things that you might be merging with your MRI data as part of your analysis. I'm going to try to keep the whole talk physics light. I'm not going to use any equations. I'm not going to use any sequence diagrams. So no matter what your background is with MRI, you should be fine to follow everything. So first, we're going to start off with MRI. What is MRI? Where does MRI signal come from? How do we generate contrast using that signal? And then a bunch of example images and sequences. So the human body is filled with hydrogen atoms. We're 70% water, and every water molecule has two, but also most of our biological molecules have hydrogen in them. And every single hydrogen atom is a single positively charged proton, and possesses a property that we call magnetic spin. So you can think of each atom as having some magnetic properties to it, and they're spinning on an axis, and this lets us do fun things with them. Those individual spins are too weak to measure on their own. We have no way of measuring the magnetic properties of a single hydrogen atom when it's in the body. But if we put those magnetic spins in a strong external magnetic field, like a very strong MRI magnet, they will all line up with that magnetic field. And the result is we have net magnetization. All of these hydrogen atoms together pointing in the same direction have strong enough magnetic properties for us to measure. In the current state, our red arrow is our big magnetic, our big MRI magnet. These are pointing in the same direction, so that is hidden right now. We don't have a way of directly measuring these when they're kind of buried behind our big strong magnetic field, but we can do something about that. We can use an RF pulse, a radio frequency pulse, and we design this pulse to match hydrogen's natural or resonant frequency. If you've ever seen like a tuning fork experiment, if you take two of the same tuning forks, you start one up and you hold it near the other one, because the air is vibrating with the natural frequency of the other one, it will also start vibrating. We have similar properties throughout physics. And so if we match an RF pulse to hydrogen's natural frequency, and we apply that at an angle to our main magnetic field, our hydrogen spins will tilt towards that RF pulse. And when we remove our RF pulse, they will start spiraling back to the longitudinal axis. So we've now taken this net magnetization and moved it into our transverse plane. It's going to start spiraling back towards the longitudinal axis over milliseconds to seconds, that sort of time scale. And this releases RF energy in the same way that we put RF energy into the system to tilt it. And that release of energy, as the transverse signal returns to the longitudinal axis, we're able to measure with what we call receive coils. So if you've seen a head coil in an MR as an example of a receive coil, has a bunch of little loops of wire in it. And when that signal passes through that loop, it induces a current. And that's how we generate our raw MRI signal. We do a bunch of other tricks like encoding things spatially so we know where that signal is coming from. And we can create an image by mapping that. But this is kind of the raw, where does our signal come from? So now we have signal. How do we get contrast? We need to be able to tell things apart in our images. How do we do that? And the answer comes back to these hydrogen atoms. Depending on what molecule the hydrogen atom is a part of and the kind of magnetic environment of that molecule, these hydrogen atoms will give us different signals. And so for each molecule or each tissue type, and depending on the MRI sequence we're using and how we set it up, we'll get a different signal back from each of these environments. And so anywhere that we have a unique magnetic property in the brain or body, we can generate contrast. And if it's not there on its own, we can do some tricks to create contrast and then measure that. And that's what a lot of our more advanced MRI sequences depend on. So this is just a little picture of an ethanol molecule to highlight that there are actually 3 different types of hydrogen magnetic environments in this molecule, and they will each give us different signals. And that holds true for hydrogen molecules attached to any molecule anywhere that we put in our magnet. So I'm going to start with our most basic of MRI sequences. You've all probably seen something like this. These are, we either call them anatomical or structural sequences because they highlight the anatomical contrasts that already exist in the brain. So white matter is there's more fatty tissues in white matter. There's different levels of water concentration in white matter versus gray matter versus cerebral spinal fluid. And so they each have kind of these innate magnetic environments that are different. And so I've got 3 different sequences here, and kind of depending on how we adjust the timings, we can highlight different contrasts. So on the left, we have what we call a T1 weighted anatomical sequence. In this sequence, our white matter is the brightest signal. The gray matter is a little darker, and our CSF is very dark. In the middle image here, we have a T2 weighted sequence. This is the inverted contrast. So our white matter is now the darkest and our CSF is very bright. On the right is something we call a proton density weighted image. You'll notice there isn't actually as sharp of an anatomical contrast. This is just a sequence that is designed to tell us the total concentration of hydrogen atoms, if you want to think about it like that, in every region. So it's just maximizing all of the signal possible. Not a lot to do directly with this, but there are some cool physics tricks you can do in your analysis using this type of sequence in combination with others. Let's move on to another popular MRI sequence that you're likely familiar with. This is diffusion weighted imaging. And here, one of the contrasts that we can use that is also innate to the body is how water is diffusing. So on the left, these yellow little water molecules, these water molecules are not in an environment with any boundaries around them, so they're freely diffusing. You can see them just kind of wandering about. On the right, we have these blue cylinders. These can represent something like axonal bundles. And in the presence of this type of boundary that the water can't freely diffuse past, more diffusion is going to happen along that direction. And so basically, depending on the tissue and microstructural environment in the brain, water is going to diffuse differently. And we can take advantage of this and generate MRI contrast based on this. So I've got 4 diffusion-related images here to show you. On the far left is a raw DWI image. Not a lot to look at here. If anyone can look at raw diffusion images and tell you much about them, they're a special person. But with a little bit of analysis, we can generate some fun images. The second image here is what we call a fractional anisotropy map. And so regions that are bright in this image correspond to kind of this second environment on the last slide. We have more restrictions, and the water is diffusing preferentially in a single direction. And so it happens in the brain that most of this restriction corresponds to white matter tracks. So basically what we end up with here is a map of the white matter bundles in the brightest regions. In the third image, what we've done is taken information about which direction the water is diffusing. We have that information as well with some processing. And by overlaying that in color on the FA map, we can now look at a single 2D slice and tell where the white matter tracks are and which direction they are in this slice. And the last image, neuroscience's favorite image, I think, based just on journal covers. This is a tractography map. By looking at a single voxel and seeing which direction the water is diffusing, and then following that from voxel to voxel, we can basically map out these tracks, then do some fun coloring, do a 3D render, and end up with an image like we see here. And this can be interesting for, you know, now that we can follow these water diffusion tracks, and these should be highlighting neuronal tracks in the brain. We can make estimates about structural connectivity between regions based on things like this, right? If we're able to find water molecules that preferentially diffuse from one location to another, we have increased confidence that they're structurally connected in some way. This is a sequence called arterial spin labeling. We use this to get blood flow measures. So on the left, similar to diffusion, a raw image that tells us very little, but with a little bit of processing. Our middle image here is what we call a cerebral blood flow map. So in arterial spin labeling, we do a fun trick. We magnetically label the blood in the neck, give it a few seconds to flow into the brain and diffuse into the smaller vessels. And then we collect our image of the brain. And so what we're doing is basically saying how much of that signal has managed to get to different regions of the brain. And this corresponds to kind of the total amount of blood flow happening in each region. So in this middle image, you can see we basically kind of have an outline of gray and white matter. Roughly, you can see that anatomy there. And that's as expected. We expect more blood flow in the gray matter, where we have a high energy consumption, and less blood flow in the white matter, where we've got these long neuron bundles that require less energy. And if you have some extra data, this third image is something called a CVR map, cerebrovascular reactivity. If you both have ASL data and you also have a measure of the CO2 level that the subject's breathing out, you could measure this with a cannula and some physiological equipment. You can get a map like this. The brightness in this map corresponds to how quickly and readily the vasculature reacts to changes in CO2. CO2 is a vasodilatant. It dilates all of our blood vessels, and so has a similar response to if your brain is activated. And so we can do this type of mapping with this sequence as well. Staying in the vessel train, if you want to see the vessels themselves, not just the impact that they're having on something like blood flow, we've got sequences for that. This is a time of flight sequence that we use to image arteries. Because blood is moving very quickly and the rest of our tissue is not, we have a contrast between movement and non-movement. And so by designing a sequence that emphasizes that, we basically end up highlighting all of our large arteries. Some of our smaller arterioles, by the time blood gets down to smaller arterioles and capillaries, it's too small to be detected in this sequence. But we end up with this kind of, this is a projection map of this data that shows us all our arteries. And so if you needed to know where certain arteries were to correlate with analysis using another sequence, you might use a sequence like this. Conversely, if we want veins, we can use a sequence called susceptibility weighted imaging. I'm going to touch on this as we get into functional MRI, but our veins contain more deoxygenated blood than our arteries, and deoxygenated blood has exposed iron atoms in the heme molecules, and this has a susceptibility effect. It basically decreases our MRI signal in a region. And so it turns all of our veins black in our image while the rest of the tissue remains bright. And so using this, we've mapped out our veins in the brain. We can do some things that don't give us images at all. MR spectroscopy is almost a precursor in a lot of ways to MR imaging. Of course, it's advanced at the same time. But this allows us to, instead of mapping out things across the whole brain, we look at a single voxel and get metabolite concentrations within that voxel. So things like creatine and choline, we're able to measure the concentration of those within a region. We also have specialized sequence for measuring more specific brain metabolites. Things like GABA and glutamate can now be measured with MR spectroscopy. So that's fun. In highlighting a bunch of different sequences for you, I also want to highlight that they have a lot of overlap and shared use cases and specialties. Here I've got 4 images for you from the same subject. This is a stroke subject. So in the top left region of the brain, on some of these images, you can see different contrast, and this is the region where the stroke infarct happened in this subject. So we basically have, some blocked blood vessels, no blood flowing to this region. The tissue around that dies and becomes necrotic. And that impacts our signal in different ways for different sequences. So on the top left is a flare sequence. This is a specific type of T2 weighted anatomical sequence. And unless you're a trained radiologist, you probably don't notice anything when you just look at this sequence. If you know the region to look at based on these other images, you may be able to see some small subtle differences, but this is basically just a highlight that strokes aren't particularly easy to see on anatomical imaging. We need more specialized types of imaging. The top right, we have a diffusion weighted imaging again. So this is highlighting the diffusion of water. And in this region of infarct, where we have dead tissue, the molecules aren't active and moving anything, there's no blood flow to this region, we have much less water movement on average than in the rest of the brain. And we end up with this bright region, which is showing areas where the water is stagnant. It's not moving, it's not diffusing as much as other regions. In the bottom right is another sequence that I've already talked about, arterial spin labeling. So this is measuring blood flow. Again, in our region of stroke infarct, we don't have blood flow. These vessels are blocked, and so we end up with this big dark region where we're not seeing any significant blood flow. The bottom left is a sequence I haven't talked about yet, more common clinically than in research. This is dynamic susceptibility contrast imaging. So this involves actually injecting a contrast agent into the blood of our subject. We give some time for that contrast agent to circulate through the blood system. And what's happening here is this particular contrast agent shows up as very bright in the images. And what's happening is in most of the healthy brain tissue, the contrast agent passes through and the brain is doing a good job of clearing that. It stays in the blood, it's not building up. But we see this bright region where the stroke infarct happened, and that's the contrast agent building up in this tissue. If it makes it that far, leaves the blood vessels and stays in the tissue, there's not active processes happening here to remove that, and so we build up and generate this bright spot. We don't use that in research as much, just because we try to avoid injecting things. It's harder to do in research than it is in a clinical environment. And a lot of research is focused on trying to move away from more invasive techniques like contrast agents, coming up with alternatives. This is a great example. If you can do diffusion weighted imaging or arterial spin labeling, you don't need to inject the contrast agent for this type of case. And the last sequence I'm going to talk about, because I'm about to do a whole section on it, is functional MRI. This is a very boring raw image from an EPI sequence that I'm going to talk about, but it has maybe one of the most interesting contrasts built into it of anything I've talked about so far. So I'm going to talk about bold fMRI. fMRI is functional MRI, so it's any technique that highlights functional activity in the brain. Bold is a type of contrast. So when we say bold fMRI, it's a particular type of functional MRI. It covers more than 99% of the functional MRI we do in neuroscience, but we can do functional imaging with other techniques. ASL, for example, it's possible to do functional mapping with ASL. I'd like to give credit to Jody Coulomb at Western University. I've based some of my slides on some of her slides. She has a great website for introducing you to fMRI if you're interested. I'm going to talk about our bold contrast, where that comes from, the type of sequence we use in functional imaging, how you might set up a basic experiment, some of the common, yay, Jody. Jody's fantastic. Some of the basic analysis steps that we use in functional analysis, and then a quick look at some advanced sequences and advanced analysis steps that you might incorporate. Some of these maybe aren't advanced per se, but just not kind of the archetypical functional MRI experiment. So what we're looking for is a method of visualizing functional activation in the brain. And we need that to have some degree of spatial and temporal sensitivity. We need to be able to tell what region is active, and we need to be able to, sample it quickly enough that we can tell from, moment to moment what things are changing. And what do we have? We have an MRI machine that can visualize net magnetic field properties. And we have active neurons that don't actually intrinsically alter the MRI signal. I want to drive that home to anyone who's unfamiliar with that. Active neurons and inactive neurons do not create different MRI signals. We need an intermediate to measure neural activation with MRI. And that's this last part. We need a link, something that changes when neurons are active, and something that we can detect with our MRI. So I'm going to introduce a concept I briefly touched on earlier called susceptibility artifacts. Here I have a very dramatic example. If you have a hair tie with a little metal clip and you don't take that off when you go in the MRI, your anatomical image will look something like this. You can see we have this dramatic geometric distortion. But the other thing I want to highlight is you can also see that we have this kind of signal void, this like dark region where we've just lost all of our signal. And when we take our hair tie away, everything goes back to normal. The reason this is important is we have a biological hair tie in our body. All of our red blood cells contain hemoglobin. Hemoglobin has these heme groups that have iron molecules, and iron is naturally susceptibility inducing. Yes, it's stunning, Alex. Biological hair ties. Why do we have hair ties flowing around in our body? This is terrible. But it's great news for us in MRI because not only do red blood cells have these iron molecules, which are naturally susceptibility inducing, but they also have two different states. When red blood cells are oxygenated, you can think of the iron molecule as being hidden, and it has no significant impact on our magnetic field. But when red blood cells are deoxygenated, when there's no oxygen bonded to these heme groups, that iron is exposed and has a susceptibility effect, which you can kind of is illustrated on the right here with these bent magnetic field lines. Here are some images from some of the earliest functional MRI experiments that were done. On the left is MRI images of vials of blood. On the right is images of a rat brain. The top of both of these is oxygenated blood. So in our vial, the oxygenated blood looks like a vial. This is a kind of cross section through a test tube, if you want to think about it like that. The bottom blood is deoxygenated, and you can see we have this weird distorted magnetic effect around this vial. In the right, the rat on top is breathing pure oxygen, and when we give an animal pure oxygen, there's not a very significant buildup of deoxygenated hemoglobin after it passes active neurons in the capillary bed. The rat on the bottom is breathing normal air, and the difference you can see between these two images is the veins are apparent in the bottom image, and this is the deoxygenated hemoglobin having a susceptibility effect in the veins where it's building up, and we can see that that's different. And this is a kind of at an anatomical level where it's very obvious, but this is also true on a very small scale. So we call this the BOLD contrast. BOLD stands for blood oxygen level dependent, which is, as Jody loves to point out, that's a little bit of a misnomer because it's actually a blood deoxyhemoglobin level dependent signal. It's more about how much deoxygenated blood there is and less about the total oxygenated blood. But what's happening at a very small scale is if you have increased neural activity in a region, it increases its oxygen consumption. And so initially the oxygen consumption increases. It takes all the oxygen out of the bloodstream. And we have a little buildup of deoxyhemoglobin. The blood vessels very quickly compensate for this by increasing blood flow dramatically to this region. It's actually an overcompensation. And the result is that we start clearing out the deoxyhemoglobin much faster. And so our deoxyhemoglobin concentration in that region actually decreases. And without that susceptibility inducing deoxyhemoglobin, our MR signal goes up. That little train there is something that is kind of missed in a lot of introductory fMRI lessons. So this is kind of a key takeaway for anyone who's new to fMRI. And it kind of explains a little bit of the disconnect between oxygen, deoxygen, and whether our MR signal is going up or down. I'm going to show you the same thing on a graph. So on our x-axis is time. On our y-axis is BOLD response. You can just kind of think of this as our total MR signal for, let's say, a specific voxel in a region that is responding to our stimulus. So our MR signal starts out at some baseline. In blue at the bottom here is a stimulus that we're exposing our subject to. You can see that just after the onset of the stimulus, we have this initial dip. That's that brief buildup of deoxyhemoglobin I mentioned. but very quickly the blood flow compensates, and we have this increase in signal as we bring a bunch of fresh blood to the region. That spikes and then levels out a little bit as we continue our stimulus. Shortly after our stimulus is removed from the subject, our signal drops back down. It actually goes below the baseline and then slowly recovers back to that baseline. And this is kind of I'm going to show you something called a hemodynamic response function later. This looks very similar, but this is on average how we expect things in the brain to respond to a given stimulus that activates the related neurons. So that's what's happening physiologically. Now we need to do something with our MRI to capture this in a useful manner. So to do this without sequence diagrams, I'm just going to compare on the left a kind of classic anatomical sequence. This specifically is an MP-RAGE sequence, which is very popular for anatomical imaging and research. And on the right is our gradient echo EPI sequence. EPI is echoplanar imaging. It's the sequence that is most commonly used for fMRI imaging. On the left, our contrasts are highlighted. Our sequence is set up to highlight anatomical contrast. So you can see we've done a good job of separating the white matter, the gray matter, and the CSF. You can see those tissue boundaries quite well. On the right, very little anatomical contrast. You might be able to see kind of where the gray matter is on the periphery. It's a little brighter than the white matter in the middle because this is a T2 weighted sequence, but you can't really make out those tissue boundaries. And that's because the contrast that this sequence is emphasizing is this bold contrast, this oxygenated versus deoxygenated contrast, which happens to correspond to active versus inactive neurons. Some other important characteristics, the image on the left, these anatomical sequences, we can often make very high spatial resolutions, 1 or a little less millimeters per voxel. On the right, spatial resolutions of 2 to 4 millimeters are more common. This particular example is 3.5 millimeters. An anatomical image like on the left may take us 5 to 7 minutes to acquire the entire brain. Stark difference on the right, we can do an EPI image of the entire brain in 2 seconds. And that's critical because one of the things we need is the ability to temporally separate the brain's response to things that we're exposing people to. And so we can only distinguish events that are as short as our temporal resolution of our sequence. So 2 seconds is pretty common. We can actually get our EPI sequences to go as fast as like a half second in some cases, but 2 seconds is very common. Before moving on to a little more fMRI, I just want to highlight some of the drawbacks of the EPI sequence. These are things that we can do a little bit to avoid, but they're kind of built-in drawbacks to the EPI sequence, the things that we need to be aware of. One of them is an artifact called ghosting. You might also hear this referred to as Nyquist ghosts or N/2 ghosts. And this just is a byproduct of the technique we use to go so fast. And we can kind of get these echoes of our main image offset depending on the field of view of our image and how big our brain is relative to that. I've got an example here on the right, which is really obvious. Your raw images, you may not even actually be able to notice this at all. But here you can see that we've got our main brain in the middle here, but then we've also got this kind of echoed ghost at the top and bottom. And one of the key problems, potential problems with this is, let's say we're looking for activation that we expect to happen in the bottom of the brain in this slice. You can see that our ghost on top is actually overlapping. So if the activation that we see is very strong in the bottom, we may actually see some false positive activation in the top of our brain because of where this ghost is located. Now, in a lot of our modern EPI imaging, the ghosting is not nearly as bad as shown here, but again, it's something to keep in mind, something to be aware of, especially if in your particular data set, you can visually see these ghosts. That's an indication that you might want to be worried or aware of possible spurious activation when you get to your mapping. Distortion is also a big problem with EPI. When we do an anatomical sequence like I've shown you before, it's pretty safe to assume that the overall size and proportions of the brain is quite similar to reality. We have some small geometric distortions in anatomical imaging, but they're not hugely significant. In EPI, we have very significant geometric distortions. This is the same subject's brain imaged with 2 different sequences where we've varied a parameter called echo spacing. This is kind of the key thing that determines the amount of distortion that we see in EPI imaging. And you can see that in the image on the left, we have kind of these horn-shaped things at the top. This is not what a brain looks like. Even on the right here, where we have a shorter echo spacing, this is half the echo spacing. You can see we still kind of have this flat top of the brain. These are both emphasizing the same problem to different degrees. Something to be aware of, we have some analysis tools that can correct for this. Depending on how you design your analysis, you might be able to ignore correcting this and just leave things in the space they're in. But a lot of the times, because our API sequences don't have anatomical contrast, we eventually need to line these up with our anatomical sequences so that we can know what regions of the brain activation is actually happening in. So we often have to correct for this to some degree. This is a very exaggerated example. You probably won't see distortions this extreme in your data, but the image on the right is not unplausible. You could easily see distortions this size. And the last artifact I'm going to talk about is something called dropout. This just has to do with the fact that in our API sequence, we wait a relatively, on MR scale, long time before we sample our data. And as we wait, our bold contrast gets stronger. So we need to wait a certain amount of time to emphasize that functional contrast. But also over time, our total MRI signal decreases. So I have the same sequence here, but with two different echo times. In the top, you can see, comparing the top and bottom, you can see that we've got regions where we have much less signal in the bottom image. This is a realistic TE for bold imaging. 30 milliseconds is very common. So depending on the region of the brain you're looking at, if it is, this is another susceptibility thing. If it's a region of high susceptibility that you're interested in, you might have to use a shorter echo time so that you maintain more of your MRI signal. You just also need to be aware that your bold contrast won't be as strong. So there are regions of the brain which are just innately more difficult to map functionally using EPI, and it's something to keep in mind. So I'm going to do very little coverage of experimental design because it's a whole field of its own, but I just want to emphasize a couple key points. The main one is that we need contrast in our experimental design. If you put somebody in a scanner and have them look at the same image the whole time, you won't be able to tell what that image was doing to the brain. We need to be able to compare in fMRI. So you need, at the very least, you need some sort of task or condition, and then you need either a different task or condition or a baseline where the subject is doing nothing, so that you can contrast what is happening in the brain during this time period to some other time period where the brain was doing a different thing. If you compare a task to baseline, you're kind of getting, what did this task do holistically to the brain? If you compare two different tasks, you might have the subject do a very similar task where you vary one key element. And now the difference between those two periods of time is going to show you what the brain's response to that key element might be. So we do all sorts of fun design when designing fMRI experiments to try and tease apart very small responses of how is the brain responding in this particular situation. And you can set these up almost however you want in MRI. You can put all your similar conditions in big blocks where you do the same thing for a long period of time. You can spread them out and jitter them, as I've shown here. You can do all sorts of things. When you delve into the statistics behind these, you'll find that different designs are better for different situations, different things that you're searching for. So do read into this if you ever end up experimenting, designing an fMRI experiment. There's a lot of detail here. I'm now going to take you through some fMRI analysis. I'm not going to talk about any specific tools or functions or anything like that. I'm just going to go over what are the most common types of pre-processing steps that we do in a majority of analyses, or at least are very common. I'm going to go through these kind of 6 things here, and then we'll revisit this map at the end. So, and I'm going to go about these in an order that is very common. There are specific cases where you might switch around the orders of these, and there's a little contention about the way to order some of these, but this is a commonly accepted order. So one thing that we do is called slice timing correction. It's very common for us to think about EPI because you'll be told like, oh, your TR is 2 seconds, which means our repetition time is 2 seconds. We're acquiring a whole brain volume every 2 seconds. We like to think about that as we acquired a whole brain, and then 2 seconds later, we acquired the whole brain again. But what is usually happening is we're acquiring a bunch of 2D slices that make up the entire brain over time. And we're doing these slices serially. So in this little diagram, we have 5 different brain slices that are making up the whole brain. But if we have, let's say, a 2-second TR, what this means is that we're acquiring those 5 slices, and it takes us 5 seconds to acquire all of them back to back. And so our slices, each of our slices aren't being acquired at the exact same time. But most of our analysis likes to assume that they are. And so slice timing correction is basically a way of us interpolating the signal at each slice so that it looks like all of our signal was acquired at the same time. So you can see here for slice number 5, even though it wasn't acquired at 0 to 4 seconds, if our TR is 2, we look at the signal of the 2 slices neighboring each other and interpolate that signal in the time course, in the time domain, so that all of the slices signal is coming at the same time. It's what we do. Motion correction. Our subjects are never perfectly still in the scanner, even if they're not doing like very obvious motions. Obvious motions are people like cracking their neck when they're in the scanner, or shifting their head in discomfort, or moving their jaw, which ends up moving their whole head. These things are very obvious, but there are much more subtle movements, which you don't typically see, but are almost always present. For example, when you put somebody in a scanner, you're laying them down on a table. You've taken the compression of gravity off of their spine, and over time, people's spines will elongate while they're laying down. So it's very common when you look at the motion analysis of your subjects that you'll see what looks like them moving up into your head coil slowly throughout the scan, basically a shift in the positive Z direction. And this is just from, they're basically getting taller as they lay on the table and their head is being pushed up into the scanner. But in motion correction, we're basically just taking the volume at every time point and figuring out what we need to do to map them all up to the same position. So we choose some volume, usually the first or the middle volume in the series, and we line every other volume up with that, so that at every time point, our brains overlap. And so the voxels correspond to the same regions in the brain for every time point. Next, we might do some sort of distortion correction. I'm giving an example here of a field map based distortion correction. So if you are wise enough to plan out your analysis in advance, you might collect a field map sequence as part of your MRI protocol. This takes 1 to 3 minutes to collect, depending on the scanner. And it basically maps out the magnetic field strength in different regions of the brain. If we know how strong the magnetic field is in different regions of the brain, and I should clarify that it's different throughout the brain because we put a brain in the scanner. When nothing is in the scanner, the field should be almost perfectly homogeneous. It's the same field strength throughout the magnet. But as soon as you throw a person in there, that messes with it because of all the weird susceptibility inducing things in our biology. And so our magnetic field strength is uneven throughout the brain. But if we know where it is uneven, and we also know some details about how our EPI sequence is designed, we can estimate what that unevenness, what kind of distortions it will lead to. We can then reverse that, apply that to our data. And so in the middle here, another example of you see those horns that I showed you before. This is kind of a stretch in the AP direction. After correcting it with a field map, we end up with a more biologically expected geometry. And that's important for the subsequent steps that we're going to do. So co-registration, I mentioned earlier, our EPI sequence, as you can see here, very little anatomical contrast. If I asked you to find, Broca's area in this image, you might struggle because there's just not a lot of anatomical landmarks. But that's what our anatomical imaging is very good for. So we like to use our anatomical images to do ROI analyses, but our functional images have all of the functional data in them. And so we line these two things up to make sure that we can use ROIs from our anatomical images in our functional images. So this just shows you the raw data on top. You can see the red line is generated from boundaries in our anatomical image. You can see that it is not overlapping with our EPI image. Our subject's position has shifted between these two images being collected. After our co-registration tool has run, these things are now in much better alignment. And so we can more safely use ROIs generated from our anatomical images to sample our functional images. Lastly, at a group level, all of your subjects have slightly differently sized and shaped brains, and they're going to be in different positions in the scanner. So we take every subject's anatomical image and line them up with some agreed upon template. This second image I have on this slide, this is the MRI template. It's an average of 152 brains scanned in Montreal. And it gives you this kind of sort of blurry template that is a representative average of a bunch of brains. And so if we line up all of our subjects' individual structurals to this template, we now have the functional, structural data all kind of lined up with this average space where we can now make group level ROI distinctions. We might do some filtering on our data. This is some spatial filtering, better known as smoothing. You can smooth to different levels. There's a bunch of reasons why you may or may not do smoothing on your data. A lot of them have to do with making our data better align with the statistical assumptions of the later stats we do. I'm not going to go into great detail on that, but something to read into if you're interested. Important to think about how much you're smoothing and why. A lot of packages will just have a default. You might not want to use that default. For example, if you're looking at a relatively small ROI, let's say you're looking at maybe a brainstem nucleus that is only 2 or 4 millimeters in size, smoothing with a kernel that's 8 millimeters might be very silly because you're taking your small region of signal that you're looking for and you're blurring it across a much larger space. You don't want to do that. So think about this. We might also do some temporal filtering. So common in typical fMRI analysis is high pass filtering. There are various reasons that you might see your kind of baseline signal drift over time. Easy example is if over the course of your scan, some of the hardware in the MRI is heating up, the signal response it gets might change over time. So you might see this like slow drift over time in your signal. And that drift isn't representative of anything functional in the brain. We want to get rid of that. High pass filtering preserves all of our high frequency signal while removing that low drift. In other specific analyses, for example, resting state analyses, you may also do a low pass filter. This gets rid of some of our high frequency noise while preserving some lower frequency noise. You may use a combination of both of these in certain analyses. Again, something to be aware of and make sure you're doing reasonable things. Yeah, so those are the common steps. That order is pretty common. There are things that are very obvious. For example, if you don't do distortion correction before co-registration, your co-registration is going to be very difficult. Ironically, in this image, you can actually see a not distorted corrected brain that has these horns. It's going to be much harder to line this up with a normally shaped brain if you haven't corrected for those distortions already. The order of things like slice timing and motion correction might be a little more contentious. There are arguments for why you may want to switch the order of those. Again, just don't blindly use pipelines. Think about what you're doing at least a little bit. We then do some basic statistical modeling. Again, there's a lot of things to go into here that I'm only going to cover at a surface level, but on our left is this hemodynamic response function I mentioned earlier. You may notice the shape of this graph is similar to that graph I showed earlier, where after the onset of some stimulus, about 5 seconds later, we get a peak in our bold contrast signal, which then drops back down, goes below baseline, and eventually levels back out. This is kind of a canonical response shape. Interestingly, a lot of people are using the same canonical HRF for all of their analyses, despite the very paper that it was derived from showing a different HRF in 2 different regions of the brain. We do have slightly different HRFs in different regions of the brain, and there are some more modern analysis tools which can take that into account if you're interested. That's fairly cutting edge stuff that's not quite as common, but one of those things that we all kind of skip over and might actually have significant impact. So this is a quick little example of general literature modeling. On our y-axis is time. So this little white line here is basically, you can think of that as the raw MRI signal for a single voxel over time. The green and red blocks here correspond to regions of time where we expose the subject to 2 different stimulus. We then basically create these modeling elements. This first one here is just a baseline. This kind of just compensates for baseline differences between subjects. And then we estimate what we would expect the response to look like to each of these stimuli. So the green stimuli occurred at this point in time, and you can basically see this HRF shape again represented here. So if a region in the brain was responding to this green stimulus, we would expect the response to look like this. Same with the red stimulus. We're then basically looking for what combination of these three things, defined by these beta weights, most closely matches our raw data, our input data. And there's going to be some remaining residual error left over. And the strength, the resulting values of these after some further statistical analysis basically give us our statistical certainty that any given voxel was in fact responding to a particular stimuli. So we do that for every voxel, and we generate a map like this. This is a single subject. We have the raw EPI data here, and then this heat map on top, where the strength of the heat map basically corresponds to how statistically certain we were that that voxel was responding to some stimuli that we input into our model. And you may do further corrections to these maps depending on the stats that are involved. Again, I'm not going to go into great detail. At a group level, if you've done this for, let's say, 5 different subjects, having done that normalization step I mentioned earlier, you then take these maps and you put them into a normal template space. You might do some sort of average or cluster thresholding, something like that. And you end up with an average activation across your entire group to a particular stimulus. This is kind of a very classic task-related fMRI experiment design and analysis. One of the critical stats things that I'm not going to go into great detail on, but be very aware of is multiple comparisons correction. In a not untypical EPI data set, a single volume may have like 500,000 voxels in it. And if you do a statistical test for every single one of those voxels, you are bound to have false positives. This left example is a very famous example now where some neuroscientists put a dead salmon in a scanner, did an fMRI experiment, did an analysis with no multiple comparisons correction, and lo and behold, we found a few voxels in the fish's CNS system that appeared to be responding to our stimuli. Fairly confident the dead fish was not responding to our stimuli. So this is just to drive home the importance of doing proper multiple comparisons corrections. Here is just another example of, as you increase your statistical threshold, you see we get less and less regions that are passing that threshold. And a lot of our multiple comparisons corrections are essentially just varying our level of significance that we accept. Although there are other things like cluster thresholding where, if you have a lone voxel that's all on its own, we're going to assume that was false. But if you have a group of voxels that are all clustered together, it's much more likely that they're related to something real. I got 13 minutes left. Yes, we're just going to keep rolling with this. Some advanced sequences that are becoming increasingly common. Multiband is very common these days. I mentioned earlier that our EPI sequence is a 2D sequence where we do slice, 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 slice until we get a whole volume. We now have sequences that are able to acquire some of those slices at the same time. So we might acquire 2 or 3 slices distributed through the brain at the same time. And if we can acquire 2 slices at the same time and we need the same total number of slices, it only takes us half as long to acquire the whole brain. So this is a method that's very popular because it has relatively small drawback in terms of our data quality, but can give us dramatic time savings, which you can then also trade off for other things. Like you could acquire at a higher resolution in the same amount of time, or much faster at the same resolution, these sorts of things. It is not completely without costs. Here's just the same subject in the same experiment done with 3 different sequences with increasing multiband factors. So basically, in this example, we're just collecting much more data in the same amount of time. So as expected, with more data, you can see our kind of statistical certainty in this visual task increases, but you simultaneously see more of this background activations. These are kind of little false positives in our test. Hopefully we can compensate for these with either some sort of multiple comparisons corrections or cluster thresholding, something like this. But again, something to be aware of. Some of this is just because of a small SNR drop in multiband imaging. Some of it also has to do with when we acquire those slices simultaneously, they really come to us as one overlapping data, which we then need to disentangle using some tricks. Our disentangling is not perfect. So sometimes we see noise on one slice that is actually coming from signal on another slice. So all things to be aware of if you use a sequence like this. Multi-echo sequences are also becoming more common. We essentially have some dead space in the average EPI. We kind of activate, we do our RF pulse, and then we wait and we collect our data. Turns out that if there's enough space in that wait time, we can just collect a whole other brain's worth of data. And if you keep going longer after that, you may be able to get, 4 or 5 volumes of whole brain data per RF pulse. Only one of these is going to be optimized for your bold signal, but your others may be useful for other things. Or as I mentioned earlier, different regions of the brain have different magnetic properties, and so might have different optimal bold timings. So that's a common thing that's done with this data. This is what I'm showing here. This is a single echo at a standard TE. This is data that we took multiple TEs worth of data, and then based on where we were getting the strongest bold contrast for each of those, we recombine that data into an optimally combined image where you can see our average TSNR has increased throughout the brain, and especially in some of these high susceptibility regions. These are regions where at our typical TE, all our signal's just gone, but using our earlier TE, maybe at 15 or 20 milliseconds, we can recover a little bit of that signal. And so sometimes this type of sequence can come for free, depending on your other parameters. You can just kind of slot an extra echo in there. Other times we have to make some trade-offs to get this sort of data, but depending on what you're looking for, it can be an interesting technique. Some advanced analysis, again, not necessarily advanced, but just not the typical kind of task-based GLM analysis I just talked about. Functional connectivity, if we have a specific region we can declare as our seed region, we can then look across the brain for voxels that have high correlating temporal patterns with that region, and then assume that those are doing something similar functionally over time. Similarly, in resting state analysis, we look more at the low frequency variations in temporal signal, and we just compare every voxel to every other voxel to see which regions appear to be doing similar things in time. You may be familiar with these resting state maps. In most subjects, if the subject is not doing anything in particular, but you do this analysis, you will find these same maps. And these kind of highlight regions of the brain that we know to be involved in similar high-level functions. We have things like auditory resting state networks, which include both the left and right auditory cortex, visual resting state networks that include all the visual areas, and we have some higher level things like executive function, etc. And you might compare a control group and a specific disease group and see variations in these. That's an increasingly common analysis type. Be aware that there's a lot of other contributing factors to resting state networks. They can have, for example, there's been a lot of things that have shown that there's high physiological explanations to these differences. So you may think that you're seeing neuronal differences and you're really just seeing like blood flow differences or something. There's a lot of messy stuff in resting state analysis, so be very careful using and interpreting it. Speaking of physiological signals, you may include some analysis steps that use externally collected physio data. Your data can be, your bold data can be impacted by anything connected to blood flow and oxygenation. So two really easy examples of this are heart rate and breathing. Your blood is not constant in the brain. There's a pulsatility to it. Similarly, your breathing throughout the scan and the impact of the change in volume of air in your lungs actually has a magnetic field impact on the brain. So if you have these measurements, you may be able to use them to disentangle the signals from your data and maybe expose stronger underlying neuronal responses. Similarly, your behavioral tasks can have some unexpected correlates. If your task is very scary, you might have a stress response that isn't what you're looking for. If you collect something like skin conductance data, you could similarly try to disentangle that stress response out of your data before looking for your neuronal signals. And your data may just have extra information in it. If you have EPI data of any type, if you connect a nasal cannula to your subject and measure their CO2 that they're exhaling, you can use the CO2 data and your EPI data to generate a CVR map. So like a small little extra add-on might get like a whole other type of data for your experiment. I'm close on time, so I'm going to finish up this section quickly. Independent component analysis is kind of this data-driven blind method. We put in our data and it searches for spatial temporal correlations in the data. I've got 3 examples here in different rows. This top row is very likely task-driven. You can see these big temporal lobe responses. Maybe this is like an auditory task, and the ICA analysis has pulled that out. This middle task doesn't seem to have any specific ROI correlations. It's just kind of all over the place. We might assume this is just a random noise component. This bottom one, this ringing around the outside is indicative of emotion. So the subject was moving at some point in this data, and that's being highlighted by this. So one of the key problems or things to deal with ICA analysis is that it blindly spits out all these components, and you need to figure out what's what. You might be able to do something useful with some of these noise components when you know that there's something specific. Similarly, you might be able to do something useful with these task-driven components, but you need to know which is which either involves a manual deciding process where you go through them all and label them yourself, which is problematic because it's hard to repeat. Or you need to come up with some data-driven way to separate these, which can be difficult, but there are attempts to do that with, for example, multi-echo data. And this is kind of just a whole domain of analysis. Everything I've talked about so far is in volume space, where you're just interested in the voxels and how they're arranged with each other. But our gray matter is actually much more of a surface. It doesn't care. So this is a little example. This is a sulcus here, a gray matter sulcus, where the surface of the gray matter is dipping into the brain. These two spots marked by the green pluses, these are very close to each other spatially. In volumetric space, these are just a couple millimeters apart. But along the cortical ribbon, they're like 14 millimeters apart, 25 millimeters apart. So these might be doing completely different functional things. But if, for example, you did a volumetric smoothing step, it may blur the data between these quite a lot. In a surface-based analysis using something like FreeSurfer, your smoothing would occur along this cortical ribbon, and you wouldn't be smoothing these distant points into each other. You would just be smoothing them along the surface. So interesting thing to consider. Given that it's almost 2 o'clock, I'm not going to delve into my multimodal section. This was just a discussion of other equipment that you can use simultaneously while collecting MRI data. And some of the considerations and advantages that you might get from that. Things like EEG, FNIRS, eye tracking, more physiological signals like I talked about earlier. These are all things you can acquire at the same time as your fMRI data and then use them to do interesting, complicated analyses. So if anyone's interested, feel free to talk to me about those. But otherwise, thank you very much for your time. And let's do some questions if anyone has them.
Oh, I'm back. Hi.
Hey.
I wanted to give you a big thank you before we go into questions. And remind people that you can ask questions in the chat or in the Q&A box. In the Q&A box, you can ask questions anonymously. If you have any like sinking MRI question that you're embarrassed to ask in front of your friends, go for it.
No question is embarrassing.
No question is embarrassing. Also want to thank you for doing a slide on surface-based analysis and fMRI. It's kind of my jam.
Yeah.
Yes,
if no one has seen it in one of Aaron's wonderful SIFTY talks, do find some slides.
Yeah.
Thank you everyone for your kind words.
Awesome. Also wanted to give a shout out. So we have enemy. Oh, wait. How do we balance new innovative methods with techniques that are more established in the field? Oh, that's tough.
Yeah, I mean, that's a brilliant question. And you'll find a lot of people who are very deeply involved in this who actually say that a lot of us are doing a bad job of balancing this. You'll find fMRI physicists who will tell you that way too many people are using not even just techniques like multiband, but even just things like parallel acceleration, which I didn't even talk about because it's so common and accepted that we rarely think about it. There are repercussions from all of these different techniques that we combine, and we very often don't think about them. Sometimes they're just, we need them to meet some minimum threshold for whatever our project plan is. But yeah, I mean, a safe way to think about it, I think, is especially like on the acquisition side, it's do as little as you can to get what you need. If you don't need a particularly high resolution for the question you're trying to answer, don't use a higher resolution. You're going to lower your data SNR. Things are going to get harder for no reason. Same thing with if you can design your task without much complication to not need as high of a speed in your EPI, like your temporal sampling. Same thing. There's no reason to push the boundaries on these things if your question doesn't demand it. It can be tempting because then sometimes it becomes a more generally usable data set later. Things like the HCP data set we look at with awe in our eyes because they're so technically advanced, and they do let us approach kind of questions that they weren't thinking about when they designed it. But sometimes that costs you in other ways, and if you're not prepared for that, it can lead to problems later. And with analysis, I would just say keep reading the papers, right? If there's only 1 or 2 papers on a technique and you're wanting to use it to answer a question that could maybe be answered with a different design and a more basic, well-validated approach, maybe you shouldn't jump right into that new thing. Of course, the new things can be very exciting and lead to incredible high-impact papers where you discover exciting new things. So it is a difficult balance. You kind of need to know your strengths as an individual and as a lab, what resources you have available to you. If you have the right people around you to do cool new things, go for it. Yeah. So this is a realignment question essentially. If we have ROIs in structural and functional space, I'll give some broad advice in that domain, which is if you're trying to get a good alignment between your functional and structural space, first make sure your functional space is good. This involves the distortion correction I've talked about. You can do that with approaches like field maps where you collect other data that you use to correct it. There is also like increasing validation for data-driven approaches. I know there was a paper not too long ago that compared using field map distortion to just using nonlinear registration with ants, and found that if your field maps are perfectly designed and everything is ideal, the field maps perform better. But if your field maps are anything less than perfect, just doing a nonlinear registration actually outperformed. I know for a while this was the default implementation in fMRI prep, which you're going to hear about later. They actually started using the ants approach by default if you didn't give a field map. I think they've actually since turned that off, but it's still available there for you to use if you want to try it out. And then the other thing that sometimes big pipelines like fMRI prep are often very good at this, but if you're doing something on your own, people will often do poorly, is you want to reduce your total number of sampling and interpolation steps. So like when you do motion correction, you're lining things up and you have to resample the data to get it into that position. And then if you align your functional to your structural and you move that, you're resampling to get it into that new space. And every time you resample, you're kind of introducing some inherent smoothing to your data. And you don't love that. So when possible, a lot of these steps, you can just get the transform information, but not actually do the resampling. Then you can stack all of your transforms together into a single resampling. Tools like fMRI prep are great at this. They just build these things up as they go and do very minimal resampling of your data. But something to be careful of when you're putting together pre-processing tools on your own.
Yeah, I have to champion the fMRI prep thing here because I feel like before it existed, these were all the recommended stamps for resampling. Like we knew what they were, but everybody had a lack of knowledge and how to do at least one of them and was skipping them.
Yeah, and even if you have a perfect understanding of them, actually implementing it with the software is brutal in some cases.
Yeah, and that was actually one of the major pushes or the major like technological breakthroughs of fMRI prep was actually making all of those pieces of software that were doing the best possible thing work together. And it was not an easy task. I got to thank Oscar for a lot of those steps. So if you're interested in learning about processing pipelines and how to actually run them, stay on this bat channel at 3:30 when we start talking about bids and bids apps and how to run FMRI prep.
Yeah,
and then
FMRI prep is a delightful tool. I would encourage you
Yes.
all to stick around to hear about that.
And then you can work with those outputs to transform and get all your ROIs into a space in a way that's standard and reproducible, and probably using best practices.
Yeah.
Yay. Okay, so I said stick around for 3:30, but I really mean also stick around at 2:15. So we're going to take a small break, and then we're actually going to come back with 3 people who are just either defending their theses, or thecal matter, or have just defended their thecal matter, to find out about the science that they did that used fMRI data over the past couple of years. What went right, what went wrong, what kinds of fMRI data they ended up using, and what it taught them, and just to generally get first a good example of what fMRI research looks like, some real examples of fMRI research and how these features are used, going from basics to application, and also to get their advice on how grad school went for them, and to just generally congratulate them for getting to this major milestone in their careers. So definitely stay on this channel. We're going to take a break, and we will be back at 2:15 for that discussion. And once again, let's give a big thank you to Jacob, because this was a great overview. We will be putting the video of this lecture in the replay section for anyone who missed it or want to refer back to it. And yes, I can't thank you enough, because this was a very nicely organized and nicely timed thing that I think will be referred to by graduate students and their PIs for a long time. So thank you a lot.
Praise. Thank you so much. And everyone's on break, but I'll stick around and chat for a little while. So if anyone has questions, feel free to put them in the chat. Or I see, actually, we got something in the Q&A that I didn't notice. Talk about quantitative MR methods. Not my specialty, but yeah, I can, I don't know if you're looking for something specific or you just wanted the overview. Quantitative methods typically refer to the majority of our imaging, and most of what I've shown you today is all relative. The absolute values of the images are irrelevant. It's just like white matter is brighter than dark matter, and that tells us something. Or the oxygenated blood regions are a higher signal than the lower, and that tells us something. Quantitative imaging methods are either calibrated in some way so that the raw values are intrinsically meaningful. So we have quantitative structural imaging, which gives you like the actual like T1 and T2 values of the tissue at each voxel. These can be interesting for a number of reasons. You can compare them across like subjects and scanners more freely than you can with relative weighted imaging. You can also, I mean, for example, if you have T1 and T2 maps of your data, you can kind of generate a virtual image using any weighted contrast. The T1 and T2 values like determine the weighted imaging and how they look. So if you have that raw information about the brain, you can generate a pretend image as though you would run any T1 or T2 weighted sequence. And then there's like calibrated and quantitative imaging in some other domains like ASL, where again, instead of just comparing like, oh, the blood flow is higher in this region or higher in this subject for this particular study, you can instead calibrate those, get specific, like the blood flow was measured at this value in these units, and then more easily compare that to literature values from other domains or across sites and different types of scanners where your weighted values might be different. And that's like a whole, and that's a growing field, and I think a very important field, because quantitative MRI has been very difficult to do for a long time. Usually in time, like the sequences are just long to acquire, so they're not practical clinically. And even in research, we tend to balk at the time requirements for some of them. But as we find ways to do these things faster, for example, some MR fingerprinting techniques that you may come across, which is like a very new novel MRI method. some fingerprinting techniques can essentially give you quantitative outputs where you can then do these crazy things, and you can do them very quickly. So these will definitely be increasing on everyone's radars in the years to come.
Yeah, I think that's a great explanation. Yes, so we will see everyone back here we'll give 5 minutes to grab some coffee, and then we will bio break. And thank you again, Jacob. We'll now leave the stage, I swear.
Thanks. Bye, everybody.